Hello friends, today we are going to study about protein sequencing part 2. In the last video, we studied about the sequencing of small peptides. Today we will study about sequencing of large polypeptides or large proteins. The length of polypeptide increases, the accuracy decreases. Therefore, it is important to break the large polypeptides into smaller fragments. So, what are the steps that are needed to sequence a large protein? The steps are, first of all, as we learned in the last video, that to sequence any peptide, the first thing is to determine the N-terminal residue. So, this step will be there for sequencing of large protein also. So, by determining the N-terminal residues, we'll come to know that our protein is made up of one polypeptide or it consists of more than one polypeptide, because each polypeptide will have an N-terminal and C-terminal end. So by knowing the residue present at the N-terminal, we'll come to know whether the protein is made up of single or more than one polypeptides. Once we know this, and if we found that our protein is made up of more than one polypeptide, the first step will be to break the disulfide bonds so that these two polypeptides can be separated. So the first step will be to break the disulfide bond. These steps are specific for sequencing of large polypeptide, but the determination of N-terminal residue is the step which is performed for both, for the sequencing of small peptides as well as for the sequencing of large polypeptides. So after determining the N-terminal residue, when we come to know that our protein is made up of more than one polypeptides, we'll break that polypeptide by dissolving the disulfide bond. So the first step will become the breakage of disulfide bond. After that, we have to cleave the proteins. Then after cleaving, we have to purify the fragments generated and we have to sequence them by Edmond degradation. After sequencing, we have to determine the order of fragments and then we have to locate the position of disulfide bonds. We'll study each steps one by one in the following slides. So first step is we have to break the disulfide bond. Now how we can break the disulfide bond? So this is a disulfide bond that is between two cysteine residues. This is one cysteine and this is the second cysteine. The bond is between two cysteine residues. So we can dissolve this bond either by oxidation or by reduction. For oxidation, we use perphoric acid. So by using perphoric acid, we can oxidize them. Here oxygen is attached, that is oxidation happened. So we oxidized them to break the disulfide bond present between them. Or we can reduce using dithiotritol. What it is doing? It is performing reduction. That means hydrogen is getting attached. But after the disulfide bond is dissolved and they become individual cysteine molecule, they have a tendency to again form the disulfide bond. Correct? So to prevent this, we have to acetylate it with the help of iodoacetate. So what we have done? We have acetylated the ends of the cysteine so that they cannot form the disulfide bond again. Otherwise, they'll again form the disulfide bond. So, to dissolve the disulfide bond, either we can go by oxidation method or we can go by reduction method. So, this how we'll dissolve the disulfide bonds so that the polypeptides will get separated. Now, the next step is cleavage of protein. The protein fragments or the large polypeptide has to be broken down into smaller fragments. How we can do that? By using various reagents. In this reagents, these are the list of reagents that are used to cleave the proteins. What are these? Trypsin. What is the cleavage point? Lysine and arginine. Wherever in the polypeptide, lysine or arginine will be there, the trypsin will cleave that bond. For submaxillaris protease, the cleavage site is arginine. Chymotrypsin, the cleavage site is phenylalanine, tryptophan and tyrosine. So like this, various reagents are used to cleave the protein into smaller fragments. Now all these from trypsin to endoprotease, these are all proteases. But cyanogen bromide is not a protease. Where cyanogen bromide cleave the protein? Wherever methionine is present. So After the breakage of disulfide bond, we have to cleave the large polypeptide into smaller fragments using these reagents. Now, the next step after protein cleavage is the purification of the generated fragments. How we can purify the fragments? By chromatography or electrophoresis. After purification, the next step will be the sequencing by Edman degradation. In the last video only, we learned about the Edman degradation method, how we use the method to sequence a polypeptide. Okay, so those who have not seen that video, I would recommend you to see that video for detailing about Edman degradation. But in short, the chemical we use is phenyl isothiocyanate. This chemical is used for Edman degradation, which will form a PTC adduct. After reacting it with CF3COOH, a derivative is formed, which can be then identified. So this is in short about Edman degradation. The full detailed explanation of Edman degradation is present in the last video. So once we have sequenced by Edman degradation, now the next step is the determine order of fragments. How we can determine the order of fragments? What we generally do is we use one protease, for example, trypsin. Suppose this is our polypeptide chain. We are using trypsin on this chain to break the chain in smaller fragments. And if two sites are present for trypsin, the chain will be broken down into 3 fragments. Then we will use other chemical, for example, cyanogen bromide. When we use this cyanogen bromide in this polypeptide chain, suppose the cyanogen bromide is having only one cleavage point, so it will break the same polypeptide into 2 fragments. Then what we will do? We will overlap this and this fragment to generate the order of fragments, the complete sequence. Correct? How we are going to do this, we'll understand with an example in the next slides. After the generation of complete order of fragments, the next step is to locate the disulfide bond. If our protein is consists of more than one polypeptide, then after this complete step, what we have to do? Now we have to locate the disulfide bond. How we can locate the disulfide bond? We have to follow the complete procedure from this step to this step. That is what we have done. We have broken the disulfide bond and then we have fragmented the protein. Then we have purified it, Edman degradation, and we have determined the order of fragment. To locate the disulfide bond, what we can do is we'll repeat the process, but this time without breaking the disulfide bond. So once we'll repeat the process, we'll get the sequence with disulfide bond. Now, by comparing the fragments obtained without cleavage of disulfide bond with cleavage of disulfide bond, when we compare these two fragments, we can easily locate the presence of disulfide bonds. Now, let's sequence a protein. So now, suppose we have an unknown polypeptide chain. What we can do is, let's first hydrolyze the complete polypeptide and separate all the amino acids. So what we have done, first we hydrolyze the full polypeptide to separate all its amino acid. After this, after separating all its amino acid, suppose we have found these sets of amino acid present in our polypeptide. A is for alanine. We found that there are 5 alanines in this particular polypeptide. We found that there are 2 cysteines in this polypeptide. So like this, these are the list of amino acids and the number is denoting the number of times these amino acids are found in this particular polypeptide. Clear? So what we have done, we have hydrolyzed the polypeptide to separate all its amino acids to know the compositions of amino acids so that we can know what all amino acids are present and how many times those amino acids are found in this polypeptide. Next, we'll react this polypeptide with FDNB, that is to determine its N-terminal residue. Suppose we found that the N-terminal residue is glutamate. Next, we are using one chemical, one protease, that is trypsin. And then we performed Edman degradation. Now, trypsin. What is the cleavage point of trypsin? Lysine and arginine. K is the one-term abbreviation of lysine. R is the one-term abbreviation of arginine. Lysine and arginine are the cleavage point for trypsin. Suppose this is our polypeptide. some set of amino acids are found, say for example, like this. This is the sequence of amino acids. So this K is lysine. As soon as we add trypsin, what it will do? Here it is lysine, so it will cleave this bond, the bond between K and B. So we'll get one fragment, this, and the second fragment, this. If correct, that means where it ends, K, because the cleavage point is K, lysine and arginine. So wherever in the polypeptide chain the lysine and arginine will be there, the trypsin will cleave that point. So the fragments will have either K or R at its end. That means the fragments have been cleaved by trypsin. So wherever it will find K, it will break the bond between K and the next amino acid. So the last amino acid will always be K and R if it is broken by trypsin. So by using trypsin, say for example, we found that 4 fragments are generated. That means how many sites are present in this polypeptide for trypsin? 3 sites are present. If this is a polypeptide, suppose here lysine is there, here arginine is there, here lysine is there. So how many fragments will get? 1, 2, 3, and 4. There are 3 points where trypsin can cleave in this polypeptide chain, and hence we found 4 fragments. Next, what we will do, we'll use any other chemical like cyanogen bromide, and again we'll perform the Edmond degradation. And this time we found that 3 fragments are generated. Clear up to this? Now we have to overlap these sequences to get a complete sequence of polypeptides. And how we can do that? First of all, if you notice, T1 ends with K, T2 ends with R, arginine, T4 again K, lysine. But in T3, at the end, there is no lysine or arginine. What does it mean? It means that this is the last fragment. If you see this example only here, this point is lysine, this point is arginine, this point is lysine. Now this fragments, wherever it will end, whatever the amino acid is present, that will be there. Here we won't find lysine or arginine at the end. Correct, because this is the last fragment. So from this, what we can analyze is T3 is the last fragment. Similarly, for cyanogen bromide, the cleavage point is methionine. In C1, methionine is there at the last. In C3, methionine is there at the last. But in C2, there is no methionine at the last. That means C2 is the last fragment. So by determining N-terminal residue, we already found that glutamate is the amino acid found at the N-terminal end. So where is glutamate here? In case of trypsin, this is glutamate, E. E is the symbol for glutamate. So T2 is the first fragment and T3 is the last fragment. So now we can start ordering them. How we can order them? T3 is the last fragment we know. So let's write it here. Now with T3 from cyanogen bromide, which fragment is matching? T2. Correct? Because in C2 also D, S, H, V, C, D is there, and in this end TK is there. So we can write K and T here. Now from trypsin again, where you can find TK at the last? T4. So we can continue here. M, P, done. So up to Y it is done. Where in cyanogen bromide you can find this sequence? In C3. See here in cyanogen bromide, this section YLIAC. Here also YLIAC. Correct? So here it ends in Y. Here we know before Y the 4 amino acids, that is ALIK. Now the next is here it ends with ALIK. In case of trypsin, where you can find ALIK? T1. ALIK is there. Before that, again 4 amino acids are there. That is GASM. Now GASM, where you can find in case of cyanogen bromide, that is C1. So before this, the C1 segment is present. So this how we can overlap the sequences generated by 2 different chemicals, and we can able to get a complete polypeptide sequence. Now, one question from my end for you all. The question is, you have to determine the sequence of a polypeptide which has the following amino acid composition. That is 1 alanine, 4 arginine, 2 asparagine, like that. So this composition of amino acid is given to you, whose sequence you have to determine. But here the questions that you have to answer are, the first is, what is the maximum number of peptides you can expect if you cleave the polypeptide with cyanogen bromide? Second, if you cleave the polypeptide with chymotrypsin. The third question is, when the analysis of the intact polypeptide has been done, it is revealed that there are no free sulfhydryl group present. So how many disulfide bonds are likely to be present? Correct? In this polypeptide whose amino acid compositions are given to you, you have to tell how many disulfide bonds are likely to be present. And the fourth question is, how many different arrangement of disulfide bonds are possible? So do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.